And so really we're able to conclude here and say that we really do believe that this is a system that we can use to embody these neurons in a virtual world to get a natural intelligence. And we called it synthetic biological intelligence. I've seen some people using the terms and some people using the terms biointelligence. It has a distinct relationship with organoid intelligence, in that I think that organoid intelligence is an extension of this work, moving to organoid specifically. But essentially what it is the chance to bring together a synthetic biological cell source that then can be used in such a way to display intelligence. And they show that in this very, very basic way by learning to move the paddle to hit the ball more often than they miss it over time, along with all those network-wide coordination effects. Of course, we did this, and people's first response, at least those in the machine learning community, was, okay, well and good, but how does it compare to reinforcement learning? Reinforcement learning can do this already, and it can get better over time than these cells. There's nothing that biology can offer that machine learning can't do. And that's not a hyperbole. Literally, people have said this to us. And we looked at it and went, but it doesn't feel right, does it? Because we as biological creatures can do a lot more than what some machine learning can do, and we can do it quicker and with less power. So maybe there is something that the biology can still offer, even something that is optimized, something like the game of Pong is optimized for machine learning to learn. Maybe there's still something that cells can offer us. And so we set out to test it. And we started off providing an image, essentially, into 3 different deep reinforcement learning algorithms. And we said, what would happen if what we did was we capped the amount of data you gave the deep learning algorithm to the same amount that the neurons responded. If we wanted to look at something called sample efficiency, how much data required to learn and get to a given level of performance. And what we found here was, in orange is our humans, blue is our mouse, and these 3 are different deep reinforcement learning algorithms. And what we found was, even when you feed far more information into the reinforcement learning algorithm, the biological neurons actually learned quicker and used fewer samples, even though their information was more coarse. Of course, some people came back and said to us, but of course they're going to learn quicker if the information is more coarse. There's something called decursive dimensionality. It means that your system that has more parameters takes longer to converge in a solution. So you have to give the reinforcement learning algorithm simpler information. And long story short, we did that, and they did even worse relative to the biology. And then people said, oh no, but really it's just sort of 2 types of information that you're providing your cells, the ball position, and that's it. Basically the ball position on an X and Y axis. And we said, okay, fine. So we did that as well. And the reinforcement learning algorithms did even worse relative to the biology. And then we went through, and there was a bunch of other ways that people asked us to redo the analysis, and we did that, and it comes out the same way pretty much every time. Biology is a very sample-efficient learning substrate. And so it was exciting to us to sort of say, hey, look, this does seem like a viable platform. It seems to have a lot of use cases. It's not meant to replace reinforcement learning, what reinforcement learning does well, but perhaps there are some things that machine learning can't yet do well, such as learning things with high sample efficiency, that the biology can do and could be useful for in the future. And so we really saw this as a platform technology. And while some things are, of course, further away than others, such as robotics and generalized intelligence, if that's a goal we can reach, other things such as for biotechnology and research, perhaps even healthcare for limited applications, are far more accessible to us. And that's quite an exciting thing to be able to pursue. And so there were a number of different future directions that we can and are pursuing right now. The ones that I'm going to predominantly focus on today, though, are the ones where we've generated a little bit of data for, just to sort of raise how these applications could come about. So for drug discovery and disease modeling, many of you here may be aware that we can currently use an induced pluripotent cell source to take patient-specific material, such as blood or skin cell, grow them into stem cells, and turn those into a tissue type, which we can measure for a drug response or for disease modeling. But despite these amazing breakthroughs, there still persists high failure rates. And that highlights the need for a more robust translational strategy. And I don't think anyone is really on the other side of this. I think everyone understands that we need to build better ways to test and model cells. The problem is that's currently done, proxy markers are the best people can use for function. for neurons. And I say that because the purpose and function of a neuron is not simply to have activity or not have activity. It's actually to process and do something with that information. And for that, you need to have these sort of closed loop, information-rich environments. And so we think synthetic biological intelligence does provide that interactive environment, and it could enable us to test these drug responses in disease models in a more nuanced way than what has currently been able. And what's really exciting, I think, about this as well is that even a small improvement over the current practices could offer a big benefit. At the moment, for novel drugs, the success rate sits somewhere around 1%. If you look at things that are already sort of proven out to some extent, that raises up to about 6 to 8%, but it's still low. So if we could, let's say for the novel drugs, we could move and just increase predictivity of a successful clinical trial by 1%, that doubles the existing outcome at the end. And not only is that good because it means we're able to, save money on clinical trials, it actually changes the profit ratio for pharmaceutical companies. And so hopefully could lead to more investment in this space and better searches for drugs to help people. Because at the moment, it's not a very attractive proposition investigating most diseases in the central nervous system because of how hard it is to get a successful product at the end. And that's just the way it works. So it could have a really big flow-on effect beyond just this, the simple, obvious effect that we can imagine if you improved it by 1%, you double the number of successful trials. There's flow-on effects from that. So to begin with, we started out and we took some cells, some NGN2 neurons, and this is a type of neuron that is nearly purely glutamatergic. That's a type of excitatory neuron. And so we took them, and people use them to model epilepsy in a dish because essentially they're all excitatory all the time, a little like something observed in some epilepsy cases. And we grew them on these multi-electrode arrays, and what we tried was to see how did they learn, and how did they learn if we applied different anti-epileptics, or what's commonly now called anti-seizure medication, at different doses. And what we found was, you can see here, this is that raster plot again I told you about. Here, each dot, just to remind you, is cells or a group of cells firing. And you can see that this is what the epileptic cultures look like, all firing all the time. Here's what it looks like if we put anti-seizure medication on, and that's something a bit closer to normal of what we see. And then what we found is that we could try different types of drugs to get a different response. We found carbamazepine was the most robust inhibitor to get a physiological effect to reduce activity, and this followed a dose-response curve. And what's interesting here also is it followed a dose-response curve, peaking at a certain level, of course, in terms of restoring learning. And so this kind of gave us this very preliminary evidence to sort of say, hey, not only can we get learning in healthy neurons, we can restore learning in a disease model of neurons. And so that was sort of a super exciting finding for us. And there's a lot of nuance here that we still need to unpack, but that's for another talk. We can also do things like just how do we understand how the neurons function better? And so we can investigate that as well, because it gives us this ability to start to investigate stuff like learning and information processing with a very high degree of control that's currently missing when we do it, say, in humans or animals. And neuroscience research in of itself is a significant area of human endeavor. And so being able to offer improvements here is also incredibly exciting and to be incredibly valuable. And it could also, beyond, as I just mentioned before, aid our understanding of how we work. It could also be helpful to help improve stuff like machine learning or neuromorphic computing that is inspired by neuroscience. And so these are some really exciting opportunities. And there's a lot we could talk about here, but I'm just going to pick one, which is something called criticality. Now, in a critical state, essentially, sorry, halfway through, need a coffee break. Criticality is basically a population activity is coordinated between two states. So you can imagine if someone's sleeping, their brain is following a very rhythmic pattern of activity. Activity of one neuron is very predictive of activity of another neuron. At the other end of the spectrum, you can imagine that if someone is having, let's say, epilepsy as an example, epileptic seizure, neurons are firing almost randomly and discoordinated. One neuron firing says nothing about another neuron firing. So weakly and strongly coordinated. In between that, there's a spectrum, and in the middle is a critical point where it goes from order to disorder. And what's been proposed is that criticality will actually act as a useful metric to maximize information transmission and capacity. And because of that, it's been robustly linked to a number of different cognitive behaviors in recent years, including in response to drugs. And one of the studies I thought was quite fun was as scores of fluid intelligence. However, because of this controversy, we thought it was something we wanted to investigate because it seems to be involved in so many different features. Maybe by trying to pull it apart, if it appears in our system, we can actually better understand what causes this metric to arise. And again, not to go into a huge amount of depth in this, but we looked at sort of the gold standard metrics for criticality and looked for concordance across all these different metrics. And what we found across all the metrics was that the systems were closer to these critical states when they were playing the game than when they were at rest. And so we can see here that the information input alone seemed to lead to closer to critical states. And that was a hugely exciting finding for us, given the role it's been found to occur in both humans and animals. But beyond that, it did also seem to have a moderate, albeit highly statistically significant correlation with performance and actually score in a way that we interpreted as, sort of said was necessary but not sufficient. So just because something was close to criticality didn't mean it would learn. But if it was far from criticality, it did seem to mean that it wouldn't learn. And so for us, we did interpret this, that criticality was an incredibly fundamental base of what would occur to a neural system when it receives structured information. And again, there's a lot of links touching down on sort of the physical processes of information that we think this ties in with. I'm not saying to go into in this talk, but it does sort of consolidate a lot of the theories and understandings of how criticality might arise, I think. Other people, of course, have talked about this as well, but I think this is sort of strong evidence to support that viewpoint. And what was also interesting was the fact that it was so fundamental that we could actually predict whether a neuron was playing a game or at rest with a 92% success rate just through looking at the network-wide coordination alone, which I think was really impactful. Now, this is just one example, and really the goal that we're interested in is this co-development of theories and tools. So criticality literature, we develop a tool that we can use to assess it. We could refine that tool to ask more nuanced questions about it, take the data, refine and repeat that. And for cognitive neuroscience, although this looks like a very standard thing, anyone familiar with the science would say, sure, this is how all science works. But for computational neuroscience, I should say, sorry, not cognitive, computational neuroscience, this is actually something that's been quite a barrier for testing. There's been limited ability often to test the nuances of the theories that have been proposed in computational neuroscience. And so I think it's a very valuable opportunity. One of the final things I'll just touch on is the idea of expanding this research into the field of organoids. I mentioned organoid intelligence before. This is some work there was a paper that we came out this year, laid out at Johns Hopkins by Ladis Manova and Thomas Hartung that we're involved with as well. And it's basically growing these more complicated 3D cultures. And the proposal is it could lead one day to more complicated and competent intelligences that we could call organoid intelligence because it's using these three-dimensional neural clusters that people call brain organoids. And we were interested in investigating the connectivity of these organoids. When you put multiple of them in a dish next to each other, you can see here, these were 3 organoids. They actually all looked roughly like this one on the right, but this is their patterns of activity. One challenge of organoids is activity isn't always where the cells seem to be. There's a lot of challenges. But we could see here that what was really interesting is when we started to look, this is just sort of one key takeaway, when we started to look at how do these organoids arrange themselves, the functional connectivity and the clustering was not only different spatially, as you would perhaps expect, but it was different in time. So the different time scale led to a different type of clustering and information processing activity. Well, we're inferring information processing activity. There's still more work to do there.